I'm very happy to be here with this international audience to share our story. Yes, I will mainly tell about our experience with the Italian rice. Of course, yes, we are a group of plant developmental biologists. We are plant molecular biologists, but we also try to use new genomic techniques to do some breeding. And so when we started to do that, we also had to start dealing with the society and with the acceptance of this technology. So yes, we are very happy that we managed to put the first rice, gene-edited rice in the field in Italy. We are also a bit sad that it was recently destroyed, but I will tell you the full story in this more or less half an hour that we have together. So I want to start to show a bit the people which are involved in this story. So of course I'm telling you this story, and but other people are involved. So this is our research group. So this is me. This is Fabio Fornara, the other group leader that shared this story since the beginning. This is Fabio now, and these are all the young, fantastic, amazing people that work in our group, and they really make possible all these things. So I think young researchers are really the protagonists of all these stories, and I hope that also our work as an activist to promote science-based decision on these techniques will give strength and more more possibilities for the future for young plant biotechnologists. Okay, so you probably have seen this slide many times. So we are in this situation now that in many places around the world, NGT plants, they are grown and they're deregulated, while in Europe, actually, the situation is not right anymore. It's under discussion more than not at all allowed. While we know that the example that we have is always Japan, that it's a country that it was historically against GMOs, similarly as Europe. But in Japan, I was talking with some Japanese colleagues also very recently, NGTs are perceived very positively, and a few plants, first were the tomatoes, but more plants are reaching the market. The fact the reason why in Europe NGT plants are still not allowed, you probably know, is that the sentence of the European Court of Justice of 25th of July 2018 decided that stated that NGT plants, since they are mutated with the techniques that doesn't have a long tradition, like other mutagenesis, they cannot be exempted by the 2001-18 law, European law that defines GMOs, which means that NGT plants, they are GMOs. At the moment in Europe, still NGT plants are considered GMOs, but things are changing for these plants. There is discussion within the European Commission has proposed new regulation, and also the Parliament has been discussing during the last months about it. And I will mainly tell a story of what's going on in Italy. So in Italy, I was witnessing in the last years like a new renaissance from Italian scientists that for many years they really they passively accepted the ban on GMOs. So Italian scientists, they were not growing GMOs anymore for many years. And when genomic techniques became really important, especially after the CRISPR-Cas development, they started to bring the discussion in a way that possibly these techniques, they would be exempted from the GMO regulation, and also possible to be grown, these plants possible to be grown in the field. So these are just, I wanted to show you that, so we are scientists, but also together with Fabio Fornara that I showed you in the first slide. We are also activists, so we have these nice t-shirts that we believe that science is a human right, which means that it's our right as a scientist to do our job. It's also a right for the people to enjoy the products of science, which are also possibly plants which can help cope with climate change and other problems linked to our environment. So science is not just a technology, it's a right. So, and this is what we did during this year. We tried several ways to try to raise attention on the topic of NGTs that should not be regulated like GMOs. And these are some protests that we did in Milan, in Bari, in Brussels also with some international scientists. We consume publicly actually rice which was made by was an NGT rice that we grew in the lab, and we cooked it and we consume it. So showing that together with usage, we did that, showing that as a scientist, we believe that in this technology, we believe that these technologies are not harmful for the people. So I don't see very well my slide anyway, because there is a banner on top, but anyway. So the situation in Europe, I think it really started changing in 2022 when there was public consultation from the European Commission to the European citizens. In Italy, many people participated. Germany was the first country, but Italy was also in Italy, many people also participated to this survey. And actually, the people which answered to the question of the European Commission, they mainly thought that that the legislation that is available for GMO is not cannot be applied to NGT. So this was also the idea in Italy of the farmers. These are the three trade association of farmers in Italy called Diretti, Confagricoltura and CIA, and the three of them, for the first time, all the three of them, so the total of the farmers in Italy, they also support NGTs, and they also think that should not follow the legislation for GMO. Of course, in Europe, if a plant is considered GMO, basically this plant is not allowed to be grown in the field, because there are only one exemption, but not many. I mean, in Italy in particular, it's not possible to grow any GMO, not even for scientific purposes. Okay, so you probably know, if you follow this subject, that 1 year ago there was this proposal for a new regulation of the European Parliament, of the European Commission, for the European Parliament, the Council, which stated that not all the gene-edited plants can follow the same rules. There are some gene-edited plants, of course, that contain bigger insertions. They are more similar like GMOs, because they really contain new genes from species which are not sexually compatible. But some NGT plants, they're actually very similar to those plants that they can originate spontaneously by point mutation, or that they can be produced by crossings. So this proposal proposed that plants which only contain very small insertions, like or deletions, that they've been called NGT1 plants, they should be considered equivalent to conventional plants. Now, of course, the discussion is open on a lot of details on this, but the main idea of this proposal by the European Commission is that the gene-edited plants are divided into two groups: the NGT1 plants that are that contains only insertions of up to 20 base pairs, and or deletions, and then plants that contain bigger insertions, or at least genes which come from non-sexually compatible plants. And so these NGT2 plants, they will require to adopt the coexistence measures which are similar to those they require for GMOs. Of course, these are proposals, but the good news is that last year different commissions of the European Commission, the the agriculture, the health and environment commission, they passed this proposal of the European Commission, and also the Parliament passed it with a little bit with a small majority, but it passed. So at the moment it still has to be discussed by the Council, and and there are a lot of amendments that will be taken or removed, but at least we have a path where we are slowly going to. Then we have to, for sure, adjust it to the full situation of Europe, but at least we have, it's not these plants are GMOs, that's it. Okay, and we were, in Italy, we're surprised because Italy, which is a country that is historically very much against GMOs, much more, it's the strictest, very strict. There is no possibility to grow anything, not for commercial or for scientific purposes. Since many years now. I think the last plants were destroyed in 2012, but since 2003, basically, for more than 20 years, no experimental fields with GMO plants is allowed in Italy. So we're surprised and very happy actually when at the end of 2022, our region in Italy, Lombardy, well, it's in Italian, I'm sorry for that, but actually try to make a deliberation in favor of starting field trials with NGT plants. Of course, this was going a bit in contrast with national regulation, because the national regulation was, was following. So if the Europe says that it's a GMO, then it has to follow the GMO regulation, and GMO regulation does not allow growing of NGT plants, not even NGT plants in the field. But 1 year ago, there was a law that passed that allowed until the end of 2024 to grow in the field NGT1 plants. This law exempts NGT1 plants from all the requirements that are for GMOs, which means that basically all the requirements that are asked for GMOs in Italy, they are, it's impossible so it's not possible to experimentation with GMOs, but for NGTs it was possible. It's now possible with this law. I think the scientists were a bit shocked after over 20 years. And actually, this is a screenshot of the last request to experiment in Italy by the University of Catania. It was a lemon tree with the transgenic lemon tree. It was from 2003, and it was not allowed. So this is means that it was not allowed. And so this was the last one, and then after there was ours. And this is the pipeline, how we can get to the field. So we have to ask, and then we have to submit our request to both the Italian and the European institutions that they take care of this type of requests. Then it's actually the Italian one which is giving an opinion, which then goes to the European, which comes back to us. So, and then there are some institutions, especially this one, which is called ISPRA, Istituto Superiore per la Protezione e la Ricerca Ambientale, which is giving a scientific opinion on the quality of your experimentation and if it's safe enough for the environment and so on. And then finally we need to, of course, go to the locals, and then finally, if it's okay, we can get a positive result. I tell you the story of the rice that we try to grow in the field. So the main problem for rice, it's everywhere in the world. I think I saw many people connected from different places. So you might not be interested in the Italian legislation, but you might be interested in the in the use of gene editing and biotechnologies in general to try to solve this problem. So rice blast, which is called the fungal blast, which is caused by this Magnaporte fungus, is a devastating disease. So this is a picture of a susceptible variety that I took in 2020 here close to Milan. Basically, you see that it's completely destroyed the plant. And there is a lot of genetic resistance to blast, but it's never enough. I mean, it's important to keep pyrimidine resistant genes, and it's important to keep inserting all the possible genetic resistance in the varieties, because blast is also, I mean, the fungus is also evolving, and the virulence keeps coming back. So basically what we did, we took this very famous locus PI21, and that it was published around 2009, so it's quite old, for conferring variable resistance in rice. So the mutant with the deletion in PI21 gene presents higher resistance to blast. This allele is not normally present in European varieties, because the breeders never used it for crossing, probably because it's in linkage with the region that is associated with lower eating quality, which means that probably plants with the bad kernels, they were discarded. And so in a collaboration with some plant pathologists from the Sainsbury laboratory, in particular Sofiane Kamoun and Thorsten Langner, which is now a group leader in the Max Planck in Tübingen. We're not pathologists, but we became expert in the CRISPR technology, and we applied the CRISPR technology to many different varieties. And so we decided to take one of the most cultivated rice variety in Italy, which also belongs to a type that is very much appreciated by cooks here, and it's called arborio, very good for risotto, and which is quite susceptible. And we mutated it in PI21 locus and in 2 more loci that were still on chromosome 4, but selected by the plant pathologist, because the proteins produced by these 2 loci are involved in the entrance of the fungus in the cell. So they bind the effectors of Magnaporte. So we created in 1 generation. We started this project already in 2017 with the idea that we wanted to put it in the field. And we created, I mean, in 1 year we had enough seeds to put in the field, and then we started applying. But of course, until the law that passed in 2023, we were not allowed to put it in the field. I think this law in Italy came not just out of the blue, but because, as I told you before, many scientists, they found themselves like activists, especially the young ones, to promote biotechnologies, to try to explain people what these things are. And we also try to change the word that we use. So we stopped using the word GMO, also because, I mean, these plants are different, so why should we call them in the same way of something that is probably banned forever? Maybe not, but I mean, I'm very worried that it will be difficult to bring them back as technology. So to improve communication, we enrolled the journalists, or someone which is doing a different job, and we explained what the NGTs were, and we said, okay, we should call them assisted evolution technologies, which in Italian sounds tecnologie di evoluzione assistita, so TEA. This is the acronym. And with this acronym, it was easy to communicate these things. So we communicated to the public, to the citizen, we communicated as a scientific community, especially the Italian Society of Agrarian Genetics was very much involved in this. And also the definition of this TEA is on the website of the society. And so we use this word for dissemination. We use this word to explain to the politicians what they were. And in the end, what we obtained was that they were accepted. People start to understand and to accept this thing. So on the 2nd of January 2024, we prepared our file to submit for to the Ministry of the Environment, Italian Ministry of the Environment, and also to the European portal, to ask if we could grow our blast-resistant rice in the field. And it was like 60 pages file. We needed to really give all the details. We sequenced the genomes of the plant, we mapped the mutations, we described a lot of things that we were asked. And finally, we went through all the, so this is the application form that we submitted. This is the first of 60 pages. And then we followed the all the pipeline. So this is what we did. And yeah, unfortunately, this we realized afterwards, but when we submitted the application, both on the Italian and the European websites, the location of the site was public, because the application is public, so everyone can download it, so they can also find the location of the site. This was not good, because many people that were in favor, but probably someone was not, so they came and destroyed the field. But this I will show you later. And so after less than 3 months, surprisingly, we got an email. I got an email with the authorization to plant the field in the location that we decided ourselves, which was a farm of a farmer which believed in these technologies, and he offered this field to the university for free. And so we had to made some signs, and then we could go there and plant our rice. So we had the support of this philanthropic foundation, Fondazione Vittoria Brambilla, which believes in innovation in agriculture, and is very active in the region, in the south of Lombardy region in Italy. And then it was just a university and this foundation, so this is our research field, which is very small. This is the plot. So we had 3 independent lines with the 3 mutations in the 3 genes that should give resistance to blast. At least they do in the lab, but we don't know in the field. And then we had the wild type control. So this is the plot, and you can see it's very small. And this is a buffer zone that we left with the variety that gets very much attacked by blasts. So we hope to reproduce the spores that they can go, and then we could see if our TEA, our NGT lines, they were less affected by blast than the wild type control, the isogenic lines. And then our idea was also to check the cross-pollination from our rice. It could go to different distances from our plots. This was our initial plot, and this is when we planted it. So this is maybe not as straight, and this in the drawing, but this was the idea, to follow the same drawing. We were asked to, so it's only 28 square meter field, and we were asked to put fences that would go also in the soil, and also with a net on top, so that also birds could not feed on our rice, and mice that might be in the field that would not feed on the plant and disperse our material. So we had our signs and the net and everything. We were very happy. The day that we planted our plants, there was a big party in this farm, where actually we had 2 senators of the Republic, Elena Cattaneo, which is also a scientist, a very good scientist, also pharmacologist, and there was also Luca De Carlo, which is a senator in the Italian Senate. So, and a lot of other, people with an important institutional role. And so we made this nice party. These are some younger people from the lab that they all joined. They all came. We took the plants from the lab. Finally, we could not believe it. We brought them outside in the real light, in the real condition. We brought them to the field. This is me very happy, smiling. And this is the, how do you call it? I mean, the assessor, I don't know how to call it. Anyway, the chief of the agricultural department of the region, which also was involved. And these are all the young scientists, Roberto De Fez, which also joined to help us planting the plants. Roberto De Fez is also a scientist, which is been involved actively also in the activist part of the dissemination of these technologies. This is a senator which also wanted to plant the plant. So there is we saw a strong support. There was a TV. It was a very nice party. We were happy, and we took pictures all together with the politicians and the young people from the lab. This is the owner of the farm, which is a farm which is 500 years old. So his family owns the farm since 500 years. But he likes tradition, but also innovation. So that's why he's promoting this type of new technologies, and he's hosting our experiment. And yes, these are more pictures from that day. It was a very nice party. We had also some journalists and influencers that joined, and a lot of in the news. So probably you got to me because you saw this in the news in Italy and outside Italy. It was a big news because it was something which happened after 20 years. We didn't even believe that it could happen, because when we applied, we thought, well, let's try. There's this new law, let's try, and we got the authorization. So there was that. And then on the night between the 20th and the 21st of June, someone, without writing an email, without asking for clarification, without noticing, without trying to open a dialogue. We are scientists. I'm a professor in the university, so I'm used to explain things to people, and I'm happy, actually. I mean, for me, it's intellectually interesting to speak with people which have different thoughts than me, to try to understand why, why they think these technologies are not good, how can we improve our communication or also our work. Anyway, no one contacted me. They just came overnight in the farm, and they chopped the rice. So they took the rice, which is in the cage now, it's in the paddy field, and the rice was chopped and destroyed. And yeah, so this went into the news a lot, even much more than the planting. So in a way, it was good, because in this way we reached even more people than before. We were in the TV shows in prime time, so where so more people that normally are not used to hear about the subject, they were reached by this news. So, and we also, I mean, okay, when I saw the field destroyed, I was depressed. But in a couple of days, we received so many emails, and also companies that offered money and offered help, and a lot of private people which offered. So in the end, we saw a strong support from all this. And so I have my last slide. I didn't check at the time. I hope I'm not too late. This is our slide, which is which shows you a picture from last week, 2 weeks ago probably, and last week actually. And you can see that many plants in the end survived, and more plants have been planted because they've been taken. These plants have been taken by the farmer, and then they made new roots. Who knows, rice is quite a tough plant, so we managed to replant some of them. So we will probably not get the results that we expected with this, I mean, statistically, with the good statistics, because we have much less plants. We will have to genotype the plants again, because some have been moved, because they've been removed and then put back. But at least we will get some data this year, and we are happy. But also, this is a new from yesterday. I told you that we had this law in Italy that allow us to have experimental fields until the end of 2024. Now the Italian Senate, they voted to extend this until the end of 2025. So next year we will be able to make another field. But our request is that the ministry does not disclose the location of the field. Otherwise, we might have these problems again. Someone could come by night and destroy everything. Okay, I'm finished. Thank you very much for listening. And I don't know how it works, if I have to answer questions now or later.
Okay, thank you for your story. Yeah, indeed, if there are questions now, you can type them in the chat. So I don't see anything yet in the chat. So yeah, you can all type whatever you ask. I was wondering, actually, were the plants proven in lab conditions to be more resistant to diseases?
Yes, we inoculated them and they had less lesions. So yeah, but we wanted to test them in the field where we had a population of different types of Magnaporte, and also the field conditions, I mean, are different than inoculation on the leaf. Yes, but we tested them in the lab and they looked more resistant.
Yeah, so they were promising plants.
Yeah, they were promising. Yeah, but of course we have more lines than this that we did. But this year we didn't want to ask for too much. For too much. Maybe next year we can make a bigger field with more lines that we have in the lab ready.
Yeah.
Do you think so? Can I ask something, Katrin?
Of course.
So I was going to ask, do you have a little regret that you had the minister there planting rice and you had the science people there, you know, that you made a big show of the plant? I think that was a problem, you think?
So do you think this was a problem why they destroyed it?
Yeah.
Yeah, well, but I don't think so, because anyway, the site was online, so anyone could see it. And of course we wanted, I mean, it was our purpose to have it in the news also, because I mean, doing something like this without telling anyone like this, we encouraged a lot our colleagues. And there are 2 more groups which have submitted the request to experiment in the field, tomato and wine, and 2 more groups which are ready to submit, that we know that we help them with the notification. So I mean, for us it was scientific purpose to test this plant in the field, but also I mean, we chose specifically these plants resistant to blast because the blast is a problem for many farmers, and I mean, for us it was very important to do dissemination, because you know, it's this is also part of our job. So in the end, these plants were, we hope they would not be killed, but it happened, and we had so much solidarity, really, from many people, like high school teachers that offered us help and money, and I said, okay, money we don't need, but we appreciate the support. We really got a lot of emails and messages.
That's good.
So
it's a good positive attitude. Yeah, right.
Indeed. There are some questions already now in the chat. So the first one is, were there any IP issues? And if so, how could you resolve them?
So intellectual property, you mean?
Yes, I guess.
So this is basic research. So this field is for research. So we didn't, I mean, there were no intellectual property issues. The variety original that we mutagenized belonged to a seed company, and they offered it for free for research. So they say, okay, you can manipulate it, you can use it, and yeah, in case in the future there might be some commercial interest on this, we can discuss it. But at the moment, it was just it's just for research, and the owner of the original variety, they agreed to give the variety to us for research.
This is more or less related to another question there about, do you have the license of CRISPR-Cas for commercialization, or you know anyone who has got the license, and how much was the licensing fee? So
no, I don't know. Also because commercialization in Europe is not an issue at the moment. I guess there might be a royalty in the end, I guess. So if there would be, but I'm not an expert, you need to ask to a lawyer, I think. But I guess if there would be a pipeline where a lot of companies and people, they make, they use for breeding CRISPR, then they might be, as there would be royalties on the variety, then there would be royalties, I guess, also to be paid for the technology. But it's just a matter of then.
Then I have something else. Were there any off-target effects? And if no, did you go for whole genome sequencing?
So we went for whole genome sequencing and we checked for the predicted off-targets. We didn't find any. Yeah, I mean, we were not, we went to whole genome sequences because actually the reason was that we wanted to be 100% sure that there would not be any residues of the transgene, because these days were transgenic plants where we express the Cas9 and the guide RNA, and then we segregate the construct. So just by chance, we wanted to be sure that nothing recombined in the plant genome, so that we would put in the field something which was for sure not GMO. So we did it for this, and we also checked for off targets, so we didn't find. And of course, there were some polymorphism compared to the original variety, but also, I mean, varieties that are not completely homozygous, so could also be because of segregation. But we went for the whole genome. The answer is this, but it was not required.
See some question on before transferring them to the soil, how are they being checked? Is there any requirement for like full sequencing or any recommendations for that?
Yeah, so this is again also we were the same question, no? So we were the first to do this kind of things, so there is no a clear rule saying you need the resequencing, you need, because it's still the notification that we did, it's still based on the GMO, so it's still requesting things typical for GMOs, like is the new proteins introduced is allergenic or stuff like that, which we didn't introduce anything, but so the full sequence it helps, but it's not at least in Italy, it's not required at the moment.
Then, I have two more questions. So one is, how many generations of selfing you had to do to get Cas-free plants?
So probably in the first generation, we already had some plants. I don't remember, because we did this plant in 2017. So we propagate them many times, probably a couple of generations to get it Cas-free, because there were multiple insertions. Yeah.
Another question that probably you may not have the answer since the field was destroyed, but someone asked, were there any yield penalties on the plants?
As far as we know, this was something that we wanted to check in the field, actually, because in the growth chamber, in the pot with the fertilizer, no, the plants, they don't look big as in the field anyway, not even the control. So we would have liked to answer to this question with the trial. Yes, it's a good question. Which software used for design guide RNA?
Yes.
I use. We did it many years ago, and we used the CRISPR-P, which is this one of the first ones from a Chinese university, CRISPR-P. It works well. We use it routinely, and it works well. It writes.
Then maybe a final question. Do you forecast that in future the pathogen would evolve to break the resistance?
Yeah, this happens all the time, so it's a run. I'm not an expert, so I don't want to give a plant pathology lecture, but yes, so we see that varieties that are resistant for the first years, and they start not to be resistant anymore. So these PI21 mutations should give a durable resistance, but we don't know. Maybe once we start introduce, and we don't know that the molecular mechanism is not known yet, how it works, but. Maybe if there would be a lot of plants with this mutation, in the end, the fungus might overcome it. There were any criminal investigation? Yes, there is the police, Italian police, that is investigating. But we had some cameras that were recording on the field, but they destroyed it before destroying the field. So they went first to the camera and and then so there, yeah, they. We thought that they would claim the destruction to explain at least why they did it, but no one did it. So, but the police is investigating because anyway, and there were people which went in a private farm at night, destroying a part of the field.